What do you think? Which animal is most dangerous to human beings? The great white shark, a lion, or a hippopotamus?
Maybe wolves? The great white shark?
I think it's lions. Wrong. The killer is not a predator. It's hardly 4 millimeters in size, but it is often deadly. In fact, this little creature can have a devastating effect. The female yellow fever mosquito, also known as the Aedes aegypti, is the greatest bearer of one of the most dangerous viral diseases in the world, dengue fever. After being bitten, it only takes a few days for the virus to break out. You will show acute symptoms such as a high fever and severe headache. And what is very typical, extreme muscle pains. It's also called breakbone fever. Especially with children, particularly in Asia and the Caribbean, there can also be complications in the course of the disease. And then it can be just as serious as Ebola and Lassa fever, where children actually bleed to death. And currently, there are no vaccinations available. This is exactly where the British zoologist Luke Alphey has found his approach. Together with his team, he began his research over 10 years ago. His goal was to effectively target the yellow fever mosquito.
So the idea was to try to improve pest control methods, to make something very specific to the particular pest, to minimize the off-target or environmental consequences. And so if we could have the insect itself be the agent of control, through mating with wild pest insects, then the insect's own biology makes that very specific.
The yellow fever mosquito is originally from Africa. However, it is now spreading across the world. It can already be found in over 100 countries. Amongst these are even European regions such as Croatia, southern France, and the Portuguese island of Madeira. Every year, up to 100 million people across the globe are infected with dengue fever. Up to 25,000 die due to those effects.
I was talking to a colleague about the use of radiation-sterilized insects for pest control, and I realized that the molecular and genetic tools that I'd been using for my very basic scientific research could be applied to the control of pest insects, so agricultural pests and also mosquitoes, and it really took off from there.
Luke Alphey and his colleagues were looking for a way to curtail the reproduction of the yellow fever mosquito without the use of radiation. And they found a solution by modifying the insect's genes.
The idea was to make a kind of genetic sterilization. So if you imagine that you want the modified males to go out and look for wild females and mate them, but for the offspring not to survive.
They actually found a way for the mosquito to eliminate itself. But how exactly does that work?
We put a gene into the insects and breed them so that the males have 2 copies. So when they mate with the wild females, all the offspring inherit 1 copy. And that 1 copy has to be enough to kill the offspring before they make it through to adult and fly off and bite people.
And this is how it works. In a laboratory, tiny amounts of genetically modified genes are injected into the mosquito eggs. The larvae on the inside absorb these in their cells. They are then copied into the genes. One single egg is enough to breed an entire colony of about 200 mosquitoes.
We have to arrange that the sterility can be switched off, and we arrange that a chemical additive, a chemical antidote if you like, given to the insects as larvae means that they are fertile, so you can breed them in the laboratory by giving them the antidote.
The mosquitoes are on a diet of tetracycline, a broad-spectrum antibiotic against bacterial infections. It prevents the mosquitoes from dying in the lab. After all, they're supposed to pass on their modified genes later on in the wilderness.
Of course, the males we release live only a few days, so they will die quite quickly, and then the gene that they carry will disappear rapidly because the offspring that carry it die.
A comparison clearly shows the effect of this gene deactivation. There is a print job in process. If one were now to insert a faulty sheet of paper into the printer, this would lead to a paper jam, and ultimately, the entire printing process would cease. In 2009, the time had come. The genetically modified mosquitoes were released in the 3 regions that were affected by dengue fever: Malaysia, Brazil, and the Cayman Islands, with an amazing result.
Fortunately, it was extremely successful. In all of the trials where we got at least 80%, usually over 90%, at one time 99% reduction in the target mosquito population. We couldn't measure the reduction in dengue, but models suggest that that degree of suppression of the mosquitoes would be enough to stop epidemic dengue anywhere.
That was reason enough for Luke Alphey to be nominated for the European Inventor Award in the category Research.
So it's a huge honor to have been nominated for the European Inventors Award. It's a great encouragement for the team, but I think even more so, it shows other people what we've done and the technical basis of this. So I think showcasing to the world is extremely valuable.
Winning at the 10th anniversary of the awards would be a demonstration of great recognition for the work with which zoologist Luke Alphey was able to save countless people's lives.